Hello, everyone. This is chapter 14, where we are introducing the idea of what is known as Mendelian genetics. Because the systems involved with genetic sorting, what we call recombination in some cases, especially when we're talking about artificial systems, we need to understand how and why genes will move across chromosomes, like for instance during meiosis, or when we're trying to breed plants and animals for specific purposes. It helps to know how these genes will move across generations. I would call this a quadrillion-dollar idea. If a really good idea in biology becomes, say, a drug or a blockbuster drug or some type of thing, I would say it's a $1 trillion idea. But in the case of Mendelian genetics, we are talking about a quadrillion-dollar idea. Virtually all of civilized history has revolved around developing plants and animals for agricultural, commercial, medicinal purposes. And so we're talking about literally the formations of at least the basic understanding of how genes move across generations has been a major factor in the development and evolution of society in human existence. So it can't be understated. So we're going to talk about how Mendel really gave us the keys to the simple processes that are now even more complex as we understand them further and further. What's really cool about Mendel and his understanding of genetic flow across generations, so we say transgenerational inheritance, if you will, is that it independently corroborates other data that we have from other experiments, or other experiments independently corroborate Mendel's ideas and experiments. That's how we develop theories and laws, is not through, shall we say, a biased validation of a given hypothesis, but an independent, like we say, corroborational approach. When we have lots and lots of corroboration, then something becomes more than a hypothesis. It becomes a theory. And if a theory is further corroborated, then we tend to call it a law. So, for example, we can actually see the process of chromosomal sorting, whether it's mitosis or meiosis, and that mechanism actually is corroborating how we see the sorting of genes across generations. So we have DNA. We're going to kind of review some of this over the next few slides, but again, show you how the pieces of the puzzle fit together. Here is a figure from chapter 5. It's showing something that we call the central dogma. And the reason we call it dogma is that the evidence is incomplete. It is a belief. That's what dogma is. It's a deeply rooted belief. Like, for instance, what is the dogma of Christianity or Judaism or Islam faith or Hinduism? There are leaps of faith, and one of the things that scientists generally believe as a leap of faith, because there's an incomplete data set, is that all living things have DNA that then is transcribed into RNA, in this case messenger RNA, and then translated into the language of polypeptides, which make up proteins. So this is known as the central dogma of biology. But it's good to have these steps in mind because we're going to be talking about them as we move through Mendelian genetics. And just a quick photo here. Mendel his system for studying genetics was the sweet pea. Sweet peas have different colored flowers. They have different shaped seeds, and so it was a marvelously simple and testable and repeatable system for finding the sort of basic rules of inheritance. And so we're going to be using some of his examples that Mendel used. So we have our DNA, which is wrapped with histones and other scaffolding proteins in a chromosome. Remember that this is actually a post-S phase chromosome. Normal chromosomes are not X-shaped, if you will. This is actually a doublet that's still attached. And so when we go into mitosis, those arms will separate. You've got a short arm and a long arm, P and Q. We'll be talking about that when we talk about loci. Within that genetic structure, you have genes, and we have thousands and thousands. We have 46 chromosomes, something like close to 40,000 genes. That means that there's at least 1,000 genes pretty much on every chromosome. So, the word gene is a functional region of DNA. It includes the pieces of DNA that will eventually encode proteins. We call those exons, okay? So we've got an exon here, and in this gene, another exon, and another exon. We also have sequences of DNA that will be clipped out when in our mRNA form. So when the DNA is transcribed, we'll make RNA, and then we will clip out these regions called introns, and you can see introns over here as well. But that's still not all that there is. There are also regulatory sequences, like for instance promoters, things that promote the transcription of DNA, promoter A and promoter B for these two genes. Again, a gene is a functional region. You're going to have promoters and enhancers and introns and exons and so forth. And we're going to talk about that in subsequent chapters. But it's good to know this going up front, because this is part of the inheritance process. As we transcribe DNA into RNA, and the RNA that is translated into protein is called mRNA, we're going to be modifying these things and adjusting them in order to make these functional proteins. Turns out that one gene, it used to be the one gene, one protein hypothesis. Now we know for sure that maybe one gene can encode multiple types of proteins, families of proteins in some cases, depending on how the RNA is processed. Every time a cell divides, we are generating a complete copy of the genome. So every cell, with rare exception, there are some exceptions, gametes are an example of this, and other very, very specialized cells like immune cells and some other things, lack a complete copy of the human genome in the case of a human cell. So the idea that whenever we have mitosis, we are replicating, and meiosis for that matter, we first are replicating the DNA. Now in meiosis, we're obviously taking half of that replicated DNA and putting it in one and half the other, that sort of thing. So, but when we replicate DNA, we're generating a complete copy of the original chromosome. Like I said, the exceptions, gametes, a few types of special immune cells, immunological cells, that sort of thing. And I've already mentioned this, so I'm not going to beat this to death, but make sure you understand the hierarchical levels of DNA organization. That is to say, we have the base pairing, we have the double helix, we add proteins, and then those proteins allow for compaction and more efficient storage. So every cell in our body contains about more than a yard, more than a meter of DNA. Given that the cell is microns, that is 1/1,000,000th of a meter, we have to have a very, very efficient way of storing that DNA, even when it's in active form. And again, one more thing to remember is that chromosomes are typically not this X configuration. I don't know why illustrators keep doing that, but a cell that's about to divide, so it's post-S phase, say in G2 and about to enter in mitosis, would have chromosomes that look like this. They generally don't look like that. In this chapter, we're going to focus on general heritability, but in subsequent chapters, as we move on through the rest of the semester, we're going to talk about why certain cells will only express certain proteins and not others. But it's still important to kind of understand why these proteins are important right now. Because proteins and anything that is expressed from those proteins, like for instance, say the protein makes a pigment, okay? The protein that makes melanin makes a cell darker, or a protein that has another product that may have a color gives cells what we call phenotypes. And in the case of your eye lens, the phenotype is that it is very crystalline because it is full of the protein called crystalline. Crystalline with an E is the property therein. Crystalline is the protein that gives the lens cells their crystal-like behavior. Not all cells express crystalline, or they would also be translucent and somewhat ceramic in their morphology. But that's an example of a phenotype, and phenotypes are driven by proteins, whether it's the proteins themselves or the products of the metabolism that those proteins are involved with. So again, central dogma, we make a protein, in this case in the lens cell of the eye, we're making this crystalline, which gives that cell its phenotype. We have a genotype, which is the DNA makeup, and we have the phenotype, which is the manifestation of the proteins that are expressed. And that's the central dogma. We're transcribing, translating, and then we get into expression and phenotype. We have to have an understanding of this, because what we do is we evaluate how genes are sorted in transgenerational inheritance by phenotype. And here's the figure 14.1a from your book. So what Mendel was doing was crossing different pea plants with different traits and seeing how the offspring sorted out. So for instance, we had a purple parent and we had a white parent. We shared pollen with the purple parent with the white, and then we saw differential expression in the subsequent generations, and they tended to follow repeating patterns of percentages. That's how Mendel got his data, was that he saw percentages of a given phenotype coming off of the original parents of the offspring that he was examining. The good thing about this chapter is that a lot of it is fairly straightforward. Make sure you have the terminology, okay? Make sure you understand what it means to be transcribed, okay? You should be able to identify it and explain what that term means. You need to understand what mRNA is as opposed to the original DNA. Is it 1 or 2 strands? Okay, what is it made up of? Understand what translation is, be able to describe it or identify translation from other processes. Can you write a short sentence that explains what translation is? Remember that cells are different. Even cells in the same tissue will express different proteins differentially, either because of conditions. Like for instance, if I take my skin and I flick my wrist here, like that, you heard my middle finger on my other hand just strike my wrist there next to the microphone. The cells now are starting to get a little bit red there. They are starting to express a different phenotype than other skin cells that are right next to them. So we have sort of generic patterns of expression of some genes, and those can not only be expressed because, say, it's a skin cell versus a liver cell versus an eye cell, but also what that cell has been through historically. That is to say, as cells are stimulated, we talked about cell signaling, the slow genetic responses, you can actually induce different phenotypes because of differential expression. So it's not only that a skin cell becomes a skin cell, it's also that the skin cell will change in response to stimuli. That's the essence of phenotype. Differential gene expression. Many genes are expressed only in certain cell types. And that's also the basis for things like cancer. For instance, a cancer cell divides and moves in ways that the normal precursor cell to that cancer would not have, and that's due to changes in the gene expression. And every protein has a corresponding gene. We do not, there is one little exception. I'm not going to get into it. There's one little exception where proteins in a cell do not come from the genes in that cell. And those are called maternal effect cells. So there are proteins in the egg which maybe that could not be synthesized by the cell itself. But that's a really, really rare exception. One thing that's really helpful in understanding how traits are inherited and how genes independently or sometimes co-sort is understanding the genetic organization of where genes are found on individual chromosomes. We have to the tune of 35 to 40,000 genes. We have 46 chromosomes. It would be chaos if those genes were just randomly assorted across chromosomes. Sometimes when we look at chromosomes, it does look kind of random, but it's just a complex and very, very highly regulated addressing system that this UROD is always found on this arm of this chromosome in this region. And so humans have tried to create a quote unquote Dewey Decimal System for identifying and locating where genes are generally found. We call that specific address, location, a locus. Locus is the Latin origin of things like the location or locale, if you talk about a town or something. So it's the specific and highly regulated location on a given chromosome where a gene will be found. Like for instance, this is chromosome 1, and this UROD gene is generally only found on this arm in this segment of this chromosome. So a locus is where that UROD gene is found on that chromosome. And like I said, there's a very complex Dewey Decimal System that humans have invented to try to identify specific loci. Now, genes have different versions, all right, and we call those differing versions alleles. Some alleles are silent. That is to say, if you looked at one allele versus another, you would see a difference in the genetic coding, but the genetic coding, because of redundancy and so forth, leads to no consequence. We call these silent alleles. Maybe minor or major changes to protein function. For instance, if we talk about cystic fibrosis, if we have a mutation and you have the cystic fibrosis gene and you get 2 copies of that gene, you'll have the disease, cystic fibrosis. Or we have what are called nonsense alleles. That is to say that they, for whatever reason, the change in the genetic structure means that protein has not been made. Okay, that the protein is absent. An example would be albinism, where melanin protein is not made at all. That means that there is no melanin. That means that they are amelanotic and you have an albino. So these different alleles, depending on whether they're silent or not silent and so forth, lead to what we call characters. That is to say, characters are another word for phenotype. Okay, well, we see variations in characters. That's due to the variations in alleles. And generally speaking, we have 2 alleles that we inherit from our parents. That eukaryotic organisms have 2 parents, if it's your typical eukaryote, and you inherit 1 allele from one parent and another allele from another. Those alleles may be identical or they may be non-identical. If a child or an offspring inherits identical alleles from both parents, that would be known as a homozygotic situation, or the offspring would be a homozygote for that gene. All right. If that offspring inherits different alleles, slightly different genetic variants of a gene, that would be a heterozygotic or situation, or the offspring would be known as a heterozygote. So for instance, we look over here, we have heterozygotic parents, but 2 of those 4 offspring in this case are homozygotic because they inherited identical alleles from both parents. Okay, one inherited one form of the allele and one inherited the other. If we have different alleles, that can lead to features, sometimes known as phenotypes. This case, we're going to be using the word character. Okay, phenotype is more of the physical manifestation. Character can be invisible but yet manifesting. Okay, a heritable feature, for instance, flower color. We can see flower color, so a flower color could be both phenotype and character. But let's say it's an enzyme inside the cell, we can't see it. We could say there's a character but not necessarily a phenotype. Okay. Traits are just another word. That's kind of an older word for phenotype and character. Okay. Genotype is the genetic makeup. So we have a genotype that leads to a given phenotype. In this case, we're talking about this is an autosomal recessive situation. That is to say, the parents have the dominant phenotype. Maybe one child out of the four shows the recessive phenotype. All right. And we have some heterozygotic things that where the recessive is covered up, and that gives us our last point. An organism's traits do not always reveal its genetic makeup. These three children would probably all resemble the homozygotic child. We'll be talking more about that shuffling and so forth and getting into Punnett squares and so forth in part 2 of this lecture. So thank you for your time. Please do read the chapter. It's important because there is additional material in the book that I can't always cover in lectures. Trying to be relatively brief, trying to give you the important highlights, but we want you to cover all of it. So thank you.